I'm going to talk a little bit about oculomotor issues and neurological behavior, thanks to our previous speaker who has already said a little bit about eye tracking and how important it is for concussions as one of the diagnostic tools. I'm going to focus a little bit on vision and neuroscience. I know you've heard a fair bit about brain mapping, so I'll go over that reasonably quickly, but I want to take maybe a bit of a different view on that in terms of, or a different angle, I should say, maybe in terms of evolution, and then get into the eye tracking, and we can talk about the clinical research and any case studies as well. So the first goal here, as I said, is to really talk about how the eye is not independent of the brain, how the retina is really part of the brain, and how the brain is so highly involved in vision and visual processing. So to start back, way back, we talk about the Earth being formed about 4.5 million years ago, about 3.5 billion years ago, we had single-celled organisms that didn't have a nucleus, and then about 2.5 billion years ago, we had single-celled organisms that developed a nucleus, and then we started to get into multicellular life form about 1.5 billion years ago. Why is multicellular life form so important and interesting to all of us? Is because as soon as you get more than one cell, there has to be a way to communicate. There has to be a way to coordinate what's going on between the multiple cells. So obviously from multicelled organisms we end up having multicelled organisms that come outside of the ocean, that develop into reptiles and then mammals, and as humans we are descendants of mammals. So along with that kind of evolutionary development, we also have evolutionary development of the brain. Right? The brain is a multicellular organism, and it really developed in 3 phases. We have a reptilian brain that was developed for survival. A lot of aspects of basic breathing functions and fight or flight instincts, and integration of sensory data. Being able to see something to move towards it as a snake and attack if that's a recognizable visual symbol, a mouse or whatever. We then developed the limbic brain that actually developed right above that, and that's our emotional brain, where we see the thalamus, which we'll talk about in a little bit. And finally, then we have the neocortex, neo being new, cortex being husk. And interestingly enough, when we develop in the womb, we develop in the same way. The brain develops in the same way. It develops first with the reptilian brain, then with the limbic system, and then on top, the neocortex, which is the new husk. So I take that line just to start off with because we're going to keep coming back to that from a neuroscience and a sensory perspective. Some of this next stuff may be familiar to you all, but I think it's good to revisit. And I'm going to start talking now about how vision, the outside in, how we take in light, and then what happens within the eye, especially the retina, and then from within the brain, and then from the inside out in 4 phases, talking about vision and brain mapping. To start off with, we know that we have light, and photon is the smallest unit of light, and that light goes through the cornea and the lens to the photoreceptors on the retina, and through phototransduction it converts into electrical currents. What's really interesting is what happens in the retina. Well, has anybody ever asked the question or thought about why doesn't the eye just send electrical signals from the rods and cones, the photoreceptors, to the brain? Does anyone know why we just don't function like that? Yes, too much information. And it kind of gives you the picture right over here as well. If you look at the slide, it's a little bit of a hint. Because you see it kind of developing down into a funnel, right? There's actually 100 million rods and cones and only 1 million axons. You remember when we had the dial-up internet? And you had to put the dial up, and then you'd try to watch a video, and the video would go along for a little while, and then all of a sudden it would stop and it would buffer, and then it would go along for a little while longer, and then a buffer. It's because the pipe that signal was going through was too small for the amount of information that was trying to pass through it. Now imagine if we had to do that as human beings and still survive.